Thank you, Nick. Fantastic. And I just have to take another moment to thank the team. I mean, they've really done an incredible job developing and maintaining and supporting the community with this software. And for many years, we did not have funding directly for this. And they all have their own projects going on. And so it's really a heroic effort on everybody's part. So thank you. And you should all be grateful to them because they're doing the work. Okay, so we are going to, hopefully everyone will fill out the survey. We'll put the link to that in the chat. But before we end, are there any burning questions that we didn't answer? Anything?
I have one more question.
Yes, go ahead.
I will assume that we can do this with SSVPs as well.
You mean file types, or what do we?
No, the ERPs.
Sorry, I was thinking graphics in my head. Yes, so a steady state visual response, is that what you mean? Yeah. Yes, there's a paper by Khan et al. Where they were looking at steady state somatosensory potentials. And I encourage you, that's a 2015 Brain paper. And they had a hypothesis about differences in healthy versus autistic children. But what they did is they did a sequence of these ERP inputs over and over and over. And that's the hypothesis they tested for generating. So yes, take a look at that. And that will give you a starting point to start to think about how to simulate that.
Thank you.
I'm just going to go through the top. Larissa.
Yes, thank you. This is great. And I was wondering if you could envision that the model could capture direct stimulation like TMS or EEG, a TMS, EEG, or transcranial electric stimulations at some point.
Yeah, so that's something we are studying right now in the lab. We're doing EEG and TMS. The way that we simulate the effects of TMS, or we have one paper where we looked at the effects of TACS, is we're not stimulating right now what is the field doing to the circuits. We're saying, can we use EEG to infer what the field did to the circuit? How did it change the firing properties of the cells? How did it change the connections between the cells? And so we tune the parameters that we have in HNN to map the response that we see from a TMS pulse.
Yeah, that sounds
so
great.
You could, yeah, and so that's how you do it right now. Ultimately, we would love to mix it with SIMNIBs and like get some fields actually activating on the cells. And I think there are some groups that are starting to think about that, but we haven't done that yet.
Okay, thank you.
Yep. Julio.
Yeah, thank you so much. This was wonderful. Yeah, actually I have some TMS-EE data and the source analysis, the time courses. I would like to try the software. So I was wondering if the instructions are in the tutorials, what kind of format I need to start playing with this? Something that
the things that we went over are in the tutorials, but there's much more in the tutorials that are distributed on the website. So I encourage you to just kind of walk through everything. And then you have a sense of, well, what am I going to do? I'm going to load in my signal, and then I'm going to start to add inputs to think about how this is generated. And then if you want to think about the impact of TMS, again, we're not building in the fields of TMS, but you can see the readout of what those fields have done to the circuits. And so you just have to kind of infer what happened to the local network dynamics when I had that pulse. Austin.
Hey, so yeah, great workshop.
Thank you.
A lot of information, but yeah, I feel like I have a good grasp on it now. So two hopefully quick questions. One, do you model any other inputs into the cells other than like the proximal and the distal drives? Like, do you have random noise being inserted as currents or anything like that? And then my question two would be, have you done any validation against like ECOG recordings specifically to sort of as like a more localized direct validation?
Yep, good questions. The first question, sorry, I just got distracted. Tell me one more time what the first question was. I remember the second was the ECOG.
Yeah, are there any other inputs to the circuit other than like
yes,
the proximal and the distal?
So in the simulations that we've been showing, I don't believe there's background noise, but in some of our publications, we have put background noise in there. So there's a drive that's called Poisson input. And so you can define noisy input to the cells. There's also a button for tonic input. So you can give injected current, like a set level of depolarization to the cells. In the simulations we were running through today, I don't think there's background noisy input. If it is, there is, it's sub-threshold. But yes, you can do that. As far as ECOG's interesting because there's no way right now to get dipoles from ECOG. The scaling, the sensors with ECOG aren't spaced at the same way that they are with extracranial. And so the inverse methods that people are using are not applicable directly to ECOG. And so what we can do is we can say, well, what are the fields looking like coming out of the distal dendrites? And that may be a closer connection to what the ECOG signal looks like than to do an inverse solution with ECOG and then say, is it counting for the ECOG? The inverse methods for ECOG just aren't the same. And the packages, as far as I know, are still not distributing ECOG source localization because the scaling is all different. But we have done some simulations, they're not published, where we say, okay, what's the local field near the surface? And that could be closer to what you're recording with ECOG. But thanks for that question. Bianca. Oh, no, sorry, that's you, Danny. I was just looking at the name.
I was wondering because we almost have been playing with the distal and proximal drives, but I guess you can also modify the parameters of the network and also of the individual cell. So I was wondering, imagine you have two regions, for example, frontal and occipital or parietal you are interested in, and you simulate your evoked potentials, okay? But I was wondering if the parameters of the network and the cell were like, if we should take into account the specific characteristics of those regions, because in frontal regions, I guess you have a bit different parameters. Do you take that into account, and where do we get the ranges of the parameters and how to play with that?
Yeah, that's a really good question. We recently published a paper on prefrontal cortex, and we were looking at stop signals responses in prefrontal cortex. And based on some of the literature talking about differences in prefrontal cortex connectivity compared to primary sensory cortex connectivity, the first thing we did was we changed the synaptic conductances in the local network to account for some of those differences. And then everything that we fit in the model was based on that slightly different network. And so it's not grossly different because it has the same structure, layer 5, layer 2, 3 connections. But the literature in frontal cortex suggested, well, this should be connected more than that. And so we took that into account by just changing those. And that was our new set state. And Nick had mentioned in HNN core, you can load in like Jones 2009. Well, now you can also load in DSPER 2024 because it's a slightly altered network model. I have to say, it didn't change things drastically, right? It wasn't like all of a sudden we got some kind of different looking ERP. The ERP looks like it does from primary somatosensory cortex. But you're right, how much the details of the connectivity structure, how much like 5 different types of interneurons and different types of pyramidal neurons, how much that matters, we have no idea, right? And so we're trying to develop a tool that can start to allow us to ask the question. And we're trying to do it in a flexible manner so that if we want to see, if I change that neuron a little bit, does that change things much? Well, let's ask the question, let's build in that neuron. And so it's, I think we're at very beginning days of really understanding the microcircuit origin of these macro scale signals. And there's a lot of future directions to go in. And you should be aware of the assumptions, exactly like what you're saying, like, well, this was constructed based on somatosensory cortex. Is it really valid for frontal cortex? Well, we can generate predictions and then we can test those predictions. Can we get a signal that looks similar? And so it's a starting point, but it's a really good question. As far as studying two brain areas, we've thought a lot about this, right? Like, let's take two little HNNs and let's connect them together. The challenge there is it's not just two areas connected together. They're connected through the thalamus. They're connected through other brain circuits. And so setting that problem up in a really biophysically principled way is really hard. And all of these softwares that are looking at whole brain interactions, they're using reduced representations of neural activity. And they're asking very different kinds of questions, right? It's more about the propagation of activity across the brain, not how did little EI cells and local dendritic currents contribute to a signal from this part of the brain. And they're all good tools, and they're just neuroscience tools. And so it's a way to ask questions, but you have to know the question you want to ask, and what's the right tool to ask that question. And I love the thought of looking at two brain areas, but we need a very good question. We need well-defined, principled connectivity patterns. And so there's a lot to think about there. Okay, before we end, I just want to mention a few things. The team is fantastic about answering questions. We have a discourse page and we have a GitHub issues page. The GitHub issues page is really about bugs in the software and things like that. The discourse page is more about community talking about the simulations they want to do. I am holding HNN office hours twice a month. You can sign up for those on our website and just pick a half an hour or an hour. And that's where, if you want to just talk about your project, how to get started, some things you're thinking, we've set aside regular time for that. So those are some ways to get help. All right, the link to the survey, can somebody throw that in the chat? Does anybody have that handy? I do here, right, actually. I'm gonna put it in the chat. It's on our Brown website, hnn.brown.edu/feedback. It should not take you more than 10 minutes. Take as much time as you want. It's really, really useful for us to understand what would have helped you more here. What do you want to use HNN for? Is it set up to answer the questions you have? All of that's really good. Oh, and thank you. Nick put the issues page there in the discussion page. Thank you. All right, everyone. Thank you again for taking the time. Thank you to the team. I hope this is useful for you and we generate some new translational neuroscience discoveries.
Yeah, thank you so much.
And thanks to Medicel.
Thank you. Welcome.
Bye.
Bye, bye, bye. Bye, guys.